great.
Okay. Hello, everyone, and welcome to this interview, which is part of a series of interviews where we speak to leaders across various different sectors in advanced therapy, medicinal products and biologics. And on behalf of the advanced therapy, oh, sorry, I've messed this up already. You see, I'm the person that's on the blue flag. Okay, so hi, everyone, and welcome to this interview, which is part of our series of interviews from the Academy of Pharmaceutical Sciences, Advanced Therapy, Medicinal Products and Biologics Focus Group. So my name is Zara Rattray. I'm a senior lecturer based at the University of Strathclyde, and I am the co-lead for this focus group. And I'm joined today with the other co-lead from the focus group, Chris van der Waal, who's based at the Cell and Gene Therapy Catapult, and Julian Braybrook from the Laboratory of Government Chemist. We look forward to hearing more from Julian and about Julian's role. So I'll hand over to Chris, who's now going to introduce Julian and cover his biography for us.
Thank you very much, Zara, and hello, everyone. Little bio then about Dr. Julian Braybrook, who joined the Laboratory of the Government Chemist in 1988 and has since then carried out a variety of roles. In June 2018, Julian became the Government Chemist and is currently Director of Measurement Science for the National Measurement Laboratory at LGC, a life science research and testing company where he is responsible for the science strategy and partnership development of metrology, measurement science, and regulatory analysis programs in support of the UK National Measurement System. Now, I'm going to stop there because I'm going to allow Julian to talk a little bit about that role, of course, later on in this interview. I will just say a little bit about the Cell and Gene Therapy Catapult as well, and who I am, and maybe Zara, you want to introduce where you are at University of Strathclyde. So the Cell and Gene Therapy Catapult, which is where I am sitting here in Edinburgh, has 3 other sites in London, Essex and Stevenage. And we are here to advance the sector of advanced therapy medicinal products, which generally includes viral delivery systems, such as Zolgensma, you might have heard of, and cell therapies, such as Yescarta, which again, you might have heard of. We have laboratories here, we have GMP facilities in the south, and we work across academia, private and public sectors. It's very interesting to speak with the National Measurement Laboratory because we do have strong aspirations to interact more closely as the years go on. I'll stop there. Zara?
Thank you very much, Chris. So at the University of Strathclyde, the National Measurement Laboratory is a key strategic partner for us, where we have the Centre for Advanced Measurement for Healthcare Research. And this is a mechanism where, working closely with the NML and NHS, which is the National Health Service, we develop collaborations across different areas, be it in the space of drug discovery or diagnostics or future healthcare and hospitals. And I'll just stop there in the interest of time and hand over to Chris, who's going to ask Julian the first question of today's interview.
Great, thanks very much. So we'll hear from Julian now. Enough from ourselves. I just want to start, Julian, if you don't mind, with a general introduction to the role of the government chemist. For example, how does the government chemist interact with the Department for Science, Innovation and Technology? And how does your strategy ensure public safety?
Okay, just before I answer the first question, Ian, it's a pleasure to be here today and tell you a little bit more about not only myself, but also about the work that I do. And as you've said already, Chris and Zara, you know, the interactions we already have and want to grow deeper with both of your organisations. So, yeah, so as you said, Chris, I've been government chemist for about 6.5 years now. There are the option for 2 5-year periods before my term runs out. So I'm clearly in that second period, and I'm effectively responsible for overseeing the statutory function of referee analyst. So what that means is effectively working to resolve any disputes that occur over analytical measurements and that particularly come about within the food regulatory environment and enforcement associated with that. So typically that might be, a port authority responsible for, that first port of entry into the country. They may analyse something. They will then, if they find a difference of opinion, if you like, that they find with the importer, then both sides have the right to access the government chemist function and the team that I have. And the aim really is to try and keep as much as possible out of court. So that function's been running for many hundreds of years now in some shape or form. So I'm very fortunate to be the current government chemist. The role also provides advice to government more generally around policy standards, I guess, and regulation based on measurement research, which is carried out either by the government chemist and team or indeed by my other functions as National Measurement Laboratory. And so a classic example of that would be, during the pandemic, it drew on my background and others, that we were able to provide that sort of advice around some of the validation of the testing that was being carried out at the time. As you said, the role's delivered as part of the National Measurement System, and that's overseen by the Department for Science, Innovation and Technology. So for those people less familiar with DSIT, then effectively this is the ministerial department that's been tasked with accelerating innovation. It's always been associated with investment and productivity through world-class science being carried out within the UK more generally. And the other thing that it's really about is making sure that new technologies that are being developed are deployed safely across the UK, preferably, and they also have a remit for driving a more modern digital agenda, which obviously we're all aware of as being a priority at the moment. The strategy that the current government chemist program focuses on really has four key themes that address that bit about food regulatory safety and enforcement. So the first one is obviously protecting consumers, and we're all aware really at the moment that the sort of global food and feed supply chain is highly interconnected and very complex. And there's a large reliance on international trade and for stocks being concentrated in a handful of countries. And we can see that, as a result of the conflict in Ukraine. It's a massive provider of grain and a number of other categories of products for the UK. And the concern, obviously, in these sort of increasingly challenging times is that there's this higher risk of supply shortages, and where that happens and where it has happened, it's also happened in Europe, because of climate conditions. Then we see increased costs to the consumer, and then that drives a behaviour within consumers that may not be totally sensible, either from the perspective of nutrition of those individuals, or indeed leaves them open to potential for exploitation from fraud and poor quality products. So we do quite a lot to, the focus is really about protection of the consumers. The second bit really is about supporting business and government, obviously, but across all the four nations of the UK. And that's really about us working closer with stakeholders involved in the food and feed sector, networks that already exist, and I guess a classic example there would be the focus really around food authenticity and, again, food fraud prevention. The third theme really is looking at trying to help build a resilient national skills base in the sector. This is a challenge at the moment, but by really trying to maximise the impact of the government-funded activity that we have and the skills and capabilities we develop, either both nationally and the way we interact internationally, then this is really about trying to get the information that we have out to people in the right way, both efficiently and timely, and preferably in the right format for them. And that good example here is our cross-government work through something called the Joint Knowledge Transfer framework for food standards and food safety. I think I got that right. It's quite a mouthful, but that allows us to provide webinars. It allows us to engage in a number of discussion type fora that allow us to share experience that we have, and to hopefully answer some of the questions that we've been asked to address. And then lastly, which is a bit more generic, really, it's about how we apply some of the UK measurement expertise to look at these bigger, wider international or global challenges. And really in the food and feed sector at the moment, it probably is more focused on the transition towards a more circular food economy. And so there are a number of projects that we have initiated under this recent programme to start to think about what are the challenges that we might be faced with in another 3 to 5 years as people embrace this sector and change the way that some of the products look now to the consumer. So hopefully that addresses things quite thoroughly for you.
It certainly was thorough. Thank you very much, Julian. That's a really good overview. Zara, maybe I can pass over to you for the next question.
Sure, thanks, Chris. So the next question I was going to ask really relates to your role as Director of Measurement Science for the National Measurement Laboratory. So can you tell us a little bit more about the various different centres that NML has and the work they undertake to improve chemical and biological measurements across different sectors?
Yeah, of course, our pleasure. So as I said already, both the government chemist function and roles and programme and the National Measurement Laboratory programmes sit within DSIT, and so that's helpful actually having there are 1 or 2 other government departments we interact with, of course, but having that ownership in one place makes it easier to have a conversation across the whole piece. And we do, being part of that national measurement system, we do have a sort of a national role to serve public interest more generally. And as I said already, the government chemist tends to address the food side of things, although coverage could be broader than that. And the National Measurement Laboratory function allows us to address the other areas. So they're very complementary. And I think by us, we have a function to set not only national thinking and direction for, particularly in the sort of chemical and biological measurement space, but we also have that role to interact as a designated institute, a national measurement institute, effectively with partners around the world. Then we can bring that international experience and what others are doing back into the UK, and equally we can take the UK experience and push that back internationally. So it's a nice balance that we have there. What do we do? Effectively, we provide the sort of validation. We term it, effectively reference methods. So a lot of people measure in a lot of different sectors, and what we're about is really making sure that the methods that are available for people to use are as robust as possible. That means we have to go back and look at the validation and uncertainty and certain factors like that. Often it can involve the production of some sort of calibration material or a reference material, depending on the quality of material that needs to be provided alongside that method, or indeed on its own. So that's really what we do, and the intention is that allows comparability between other people measuring in the same space. that helps because of the interactions we have internationally to allow us, allow the UK and trade to happen more freely, because a measurement in one place can be accepted immediately readily elsewhere in the world. And it's only where you have that dispute that then we go back into other challenges. We do have some what we term sovereign, UK's measurement functions that sit very much within our space. So a lot of people that we have interacted with and do interact with will know us for nucleic acid quantitation on the more molecular biology side. On the chemical side, there's a lot around chemical purity, setting of the purity of chemicals, and that's fundamental for us to make those reference materials at the end of the day. And equally in some of the emerging areas that are useful for nanotherapeutics and things like that, and for particle analysis in water, we have some specific traceability for nanoparticle number concentration. So those are some things that very specifically sit with us, but we work more generally across that chemical, molecular biology, and indeed cell biology space, and I'll probably come on to that a little later. I suppose we'd say our expertise and advice, the intention is for people to rely on that and to have that traceability back to it, and I think we do that. The intention is to help our stakeholders address some of the current challenges that they have. So certainly for smaller companies, often they don't have access to some of the equipment that we have, and we can offer that to help solve some solutions for them faster. That might speed up their product pipeline. It may solve a regulatory issue or something like that. But I mean, we work with all sizes of companies across effectively sort of three areas: the life sciences, and that will be from diagnostics to I suppose what we term traditional pharmaceutical therapies, but also into some of the complex biopharmaceutical therapies that we interact with, both you are and also Chris, and organizations associated with that. We also work in some of the what we term green industries, in the environmental sector, and of course, as we've said already, in the food sector. And it's all about getting that measurement to underpin standards, and those can be physical standards, or they can be documentary standards, guides, etc. And that feeds through to regulation and skills and things that we've talked about already. I think I've taken a very different view over the last few years to our strategy for the NML as well, and I think it makes a lot of sense, and we're seeing the fruits of these enhanced strategic collaborations. But so we, as you can sense already, we have some core platform technologies, particularly mass spectrometry, the trace nucleic acid analysis, and flow cytometry, covering that sort of chemical, molecular and cell biology space. But clearly there are a lot of other techniques out there. And we probably haven't got the capacity to handle all of those in for the size of organization that the NML is. And so working with these other partners is absolutely critical. So the policy has been to look for supplementing these core platforms that we have with strategic technology, or indeed sector-based partnerships. And those we've looked around the UK, who offers those, where they sit in terms of the UK, if they're in an innovation district, what the focus of that is, what support there might be locally, and that's really what it's about. It's not about only supporting us. It's absolutely about getting our sort of measurement capabilities back out to the different parts of the country, the different entities and organizations that sit within those parts of the country. So enabling effectively, I guess, local application and impact to be made at source it can't all be done from a central facility sitting anywhere in the UK nowadays. I don't think that's the way forwards at all. And also, as I said already, about enhancing, skills that sit. So if we can transfer some of our thinking into day-to-day deployment elsewhere, then I think we've fulfilled our function. As you've mentioned already, you're familiar with, because we're working closer and closer together. We have strategic relationships with University of Strathclyde. We'll go on to perhaps talking about the University of Leeds a little bit more in a moment, but we also have the interactions with the University of Belfast and University of Liverpool, and between those four, we look at measurement research for health translation with yourselves at Strathclyde, particularly clinical diagnostics, again with Strathclyde, but also particularly with Leeds. Also broader medical technologies with Leeds because of where they sit in the country. They've been a heartland of medical technology development for a number of years. Agriculture, which feeds back into the food integrity with Belfast, and some of these, as I said already, novel or the transition to novel products in the food space. And then sort of pandemic response and nanotherapeutics with Liverpool. So I think it's really exciting times for us, and as I said, we're really beginning to see the benefits of building up these relationships, not only with academic institutions, but also they often have or they will have a number of interactions locally with industry, with the National Health Service, has already been talked about, and so we can exploit all of that through these types of collaborative activities.
That sounds great, and thank you very much for the response there, Julian. And we're already quite seeing good benefits from the various different centres that the NML has nationally. For example, we're interacting with University of Liverpool and University of Leeds to establish and build new collaborations. So it's really contributed to building quite a good critical mass and developing new collaborations even within the academic sector as well as between academia and industry. So it's played a very critical enabling role in that sense. Good.
So excellent feedback, and I think the current government, obviously we just had a budget, and I think the sense coming from the government already is about how we make more use of these sort of critical centres, and so I think it's really good. We don't want to operate just individually with the centres. It's about how we can join them up and how that then creates, as you said, this sort of hub network environment. So very good feedback. Thank you.
Okay, thank you very much, Julian. So I'm going to hand over to Chris, who has another question for you.
Thank you very much, Julian and Zara. There was an awful lot you said there, Julian, around measurement, and this is why this interview is so exciting, because of course pharmacists, pharmaceutical scientists are involved in measurement in the laboratory day in, day out. We have the British Pharmacopoeia, we have monographs in that British Pharmacopoeia, of course, that speak to measurement throughout. And I think this is why in this next question, it'd be interesting to understand, going from small molecules to biologics was already quite a big jump over the last 20 years. We've seen those now well established in the clinic. And the next jump is going to be, as it already is happening, into the advanced therapies. And from a pharmaceutical scientist viewpoint, it'd be really interesting to learn a little bit about what the NML are doing in Leeds, the Northern Cell Metrology Hub, as it's called, I believe.
Yes.
And how you think that will support measurement in the cell therapy space.
Yeah, no, happy to share some thoughts around that. So as I said already, the whole area of Yorkshire, but particularly the Leeds City region, has really been a centre in the UK for a while now about healthcare research and innovation. I think like a lot of parts of the country, they've also looked more internationally to see how they can compete, and certainly Leeds, is now very much a health technology startup hub. So it was really interesting when I was thinking about these strategic partnerships. What was really critical was to look at not only what was there, but what we could interact with in a complementary way. So, I think you've summarized very neatly the change that's happening in a number of these areas, looking at therapies for the future. And so taking our sort of cell metrology capabilities, and I'll talk a little bit more about that in a second, to the university environment, but setting it actually not in the university. Here in Leeds, we set it in Nexus, which is an innovation hub in its own right. It allows a number of businesses to come together, not only to work together in the hub, but also to interact with the university and indeed with Leeds Teaching Hospital NHS Trust. So I think that's what I was trying to talk about in terms of these strategic partnerships. It's about really trying to create a stronger centre from each of the individual components being brought together. 2 + 2 = 5. Well, here it's 3 plus whatever. It's that sort of concept where we get a bit more out of being able to operate together. So that's sort of why we picked Leeds. As I said, we have a number of interactions across the UK with all other centres. So it's not that everything is focused in one particular area with one particular centre, but it does allow us to think about critical mass and getting more out of what we do through these types of activities. And as I said already, the main focus is around clinical diagnostics, but also some of the supporting medical technologies. So the focus for the hub initially, has really been about looking at the building blocks, I guess, that allow confident measurement around what are known as the critical quality attributes of cells. So you'll be very familiar with that, obviously, Chris. But so what are the cell characteristics that we can measure, that we understand what that should be measured, and how do those change, particularly during some sort of manufacturing process, or from collection at source, which is actually us as patients, as individuals, right the way through something more like a traditional pharmaceutical pipeline, but bearing in mind that cells are very, very different. Chemistry is not simple by any means, but relative to a biological cell, and a population of cells all doing slightly different things at different times, talking to each other in their own particular way, it's a very different challenge. I've often likened looking at a cell as if you think about a castle keep, a tower, you've got your slits that traditionally the arrows were fired out of. Well, if you look from the outside in, effectively from a measurement perspective, then you're looking at this slit. So you look at if you're analysing a cell, you're looking at a cell at a particular moment in time with a particular analysis, and then you go a little bit further around the corner and you've got a different perspective, and of course in that time the cell's possibly changed in some way. And so the whole concept of measurement is really a bit different, and actually is quite challenging conceptually to think about how you can learn about control. I think the key thing to take away as well is that if we really want to understand true biological variability, we have to separate as much as possible the variability in the methods that we're using to look at those particular entities. Here cells, or cells plus a combination of other things. So that's really what it's about. I think it's trying to get back to those sort of quality assessments of cells, and they may be physical, they may be chemical, they can be biological, they may even be microbiological at certain points in a pipeline. But I think it's about really being able to try and control as far as possible, and understand those measures in at an appropriate limit, within a certain range, probably distribution as well, and it's all about trying to make sure that the product at the end of the day is of the right quality, it is effective, it's efficacious, and is safe.
Yeah.
So I think that's where we're starting. We're still relatively early days, but the interactions that we already have, with yourself, and I was on a call just the other day with colleagues down in the London part. We've already just initiated a project with yourselves, based at the Northern Cell Metrology Hub in Leeds, to look at enhancing flow cytometry capabilities, and our capabilities in handling some of the different cell types that are coming through as well. And why do this? Well, I've hinted about the quality and safety and function, but it really is just to be able to improve biological manufacturing of these complex therapies. What do we tend to think about as these attributes? Well, I guess for people less familiar, then the first thing probably is, are we sure we've got what we think? So an identity type thing, and is that exactly what we want? Obviously quantity, because at some point we will be dosing, so we need to know exactly how, or as closely as possible, how much of the efficacious element that we have within a certain dose. Obviously purity and sterility, certainly at release of these compounds, and of course these products are very different because they're going to largely come out of a system and go straight into a patient, so we need very different techniques at the back end of things to be able to give more certainty about sterility. They often have to be viable, and we would look at markers of sort of biological activity and things like that to give us some influence, an indication of function. There are others in the sector, particularly MHRA at South MIMS, the XNIBSC labeled functions, which look at that biological function specifically. So we interact with them to enable that to happen. And I think it's fair to say, there is still room for improvement on our understanding about these types of products, and particularly how we can measure them, as I said already, through these sort of product development pipelines. And effectively by doing this, I think we can improve the decision making that's done at certain points. That can streamline the process. It can certainly give us more information in terms of sort of assay parameters that come back, if we can learn how to feed that back into the system. And obviously data and all the sort of tools that are out there to enable us, the sector, to do that. I think those are areas where we're still learning exactly how to apply that. But if we can get these sort of more uniform or agreed acceptance criteria, then I think we will have much better and faster production of biotherapeutics going forwards.
Yeah, that's a very insightful answer, Julian. Thank you very much. I think from the viewpoint of a pharmaceutical analyst, you said precisely the right things there in terms of pointing them in the direction of new measurement techniques. You mentioned flow cytometry, viability, even cell counting. I realized when I first entered the biopharmaceutical industry isn't straightforward.
No, I know.
Trips people up.
Not a lot of things people take for granted, and it's not that it's just understanding that variability that exists. It's not that we can't count. It's just, well, that's fine, but if you're trying to prepare a very precise dose, well, what's the element of uncertainty, variability we can have around that before it makes a difference. It's not about driving everything to the nth degree. Often, we're challenged on, well, you're just trying to get the best method. Well, I think we are trying to get the best method, but the best method that's actually fit for purpose. And that's something, I think that's a really important message for people when they think about how to interact with us. It's about improving things, but to the right degree, in the right place, and at the right time. It's not always about just having a super method that, we can all have super methods that are actually only applicable in one place.
No, that's very well said. I absolutely. I think, Zara, maybe this is worth you asking maybe a closing question here around that area.
Definitely.
Could I hand over to you? Would that be all right?
Sure. Thanks, Chris, and thanks, Julian. So obviously we've covered pretty much the traditional face of pharmaceutical analysis and the novel and emerging side and the future, essentially, of pharmaceutical analysis in the context of self-based therapies. And we've got quite a diverse audience that are going to be tuning in to this interview today. And they will be from different sectors and they will be at different career stages. So in terms of what the National Measurement Laboratory has to offer, what would you suggest are the ways of reaching out and engaging with the National Measurement Laboratory, and what career opportunities are available within the NML and that you could share with our audience?
Happy to do so. So I think, hopefully, from what I've said already, you'll understand that we are all about collaboration. We have a function and we have to do certain things to enable that. Collaboration, as I've said, really is the way forward now everywhere. By virtue of that, we are open to working with anybody and everybody, in the areas that, obviously are deemed priorities at the time. There are various ways people can engage with us. Sometimes we'll reach out, but I think, we're certainly open. Anybody can contact me or any of my colleagues we're based in Teddington presently. We are moving, which is a bit of a challenge, moving the whole laboratory and not only the GC and the NML, but also functions we have for MHRA on the pharmaceutical testing side, from a chemical perspective at least, down to Guildford and the Surrey Research Park. So plenty to do at the moment, but that also will create opportunities for individuals to work with us and to hopefully join us if they would like to do that as part of their development of careers. I think possibly the best way is if I just give some examples. Often when we if we're tasked with doing something for government, we will reach out and engage with organisations in that area, and probably the first way of interacting will be very much on a sort of an in-kind basis. A lot of the areas we've talked about already, the only way we can get good measurement is if we get the right sort of samples from people. So, that can be the first sort of point of interaction. It doesn't have to be just sample exchange, but that makes sure that what we're doing is relevant, and it builds that initial relationship, and then hopefully we can, take that organisation in, whether it's a small company, or a hospital or an academic interaction, through a project and then into multiple projects and interactions, and then at the end of the day we may end up providing some services to the companies at the end of the day. We have a number of schemes that we operate on behalf of government that allows interaction with particular sectors. So there's one that's already set up to work with NHS to look at some of the challenges that biomedical scientists are having. There's interaction through Innovate UK for looking at these early challenges that small companies have where they want access to particular equipment. So again, that can be another way of interacting. It's pretty flexible, and I think that's also something that I've been very keen on making sure we're not, we have a range of different ways of interacting with different parties. It doesn't all have to be on a contractual basis, but it's just setting the right parameters that everybody preserves those sort of boundaries that's relevant for people. We work across technology readiness levels, so I guess traditionally we've always operated at trying to get that translationary bit from academia to industry right, but as I said already, often we get far more involved now in some of the post-market surveillance of some of the diagnostics and medical technologies, just to make sure that they are functioning once they're being used routinely as well, because often that's where the real data comes back to say it's working well, or there may be a challenge in a particular aspect. In terms of individuals, then through some of those interactions, particularly with academics, that's a good way for people to get to know us and us to get to know them, and because of those partnerships we have exchanges, and the relationship we have and the post that you particularly have allows some of that to happen. So there's an example, but as I said, we work across traditional chemistry from small molecule to large molecule, in different regulatory sectors. We work in inorganic and organic, small molecule, large molecule, protein-based studies, molecular biology. Nucleic acid measurement is the key for everything, but also a lot in some of the genomic technologies now that are coming through, and the interaction between the two, and then in cell biology. So the range of opportunities is really large, and in fact, the staff we have are possibly a bit more national than we've had in the past, but that's really good for the UK. But we do have a number of international or people from a more international background working for us. And because of these interactions, people that do join, we can provide opportunities for them to have further enhancement to their careers if they join, really early in their career. Often that will happen in some of the areas like MHRA. For the NML and GC, it's obviously a little bit perhaps more advanced, but we would certainly take people with degrees and masters, more than we ever used to. A lot of our staff have PhDs, but if they join early, we can also work with the academic sectors and the relationships we have to enhance their careers through other routes, if that's what we both want. So I think perhaps a very generic answer, but I think it just reflects we have a very different approach, a very open approach, and have quite a range of activities and areas that we operate in. So there are good career opportunities.
Thank you very much there, Julian. There are definitely a large and diverse range of opportunities, be it whether you're in academia, starting out in your career, or further advanced, working within the academic or industry-based sector. And I've also certainly seen examples, for instance, where with the analysis for innovators, SMEs can apply for funding and joint funds with the NML, which then gives them an opportunity to also solve some of their grand challenges in measurement science. So yeah, the range of opportunities, I suppose, and possibilities are quite diverse and endless there. But you did a great job trying to pull all that together using different examples.
Good. Hopefully I did.
So thank you so much again for joining us for this interview today, Julian. We really appreciate your time, and I'm pretty sure that all the insights that you've provided today will be of great interest to the Academy of Pharmaceutical Sciences and our audience that will be listening to this interview. So thank you very much for joining us once more.
No, it's a
Thank
pleasure.
you very much, Julian.
Yes, thank you. Bye for
Thanks.
now.
Thank you very much.
Okay, I'll stop the recording.